Hello, this is Mrs. Murdock, and I'm going to be going over how exactly to relate crossover frequency to mapping the distance between genes. So once you have figured out, looking at your data or doing your chi-square analysis, that you have two different genes for two different things that are linked on the same chromosome. Once you've figured that out, the next thing they might ask you to do is to actually come up with a numerical distance between the two genes on the chromosome. So you might think, oh gosh, that's going to be really hard. It actually isn't very hard. It's just a matter of understanding how to use the data and how to combine the data understanding with the understanding of the physical chromosome, and going back to meiosis and what chromosomes do during meiosis. So for example, number 3 here, body color can be gray or black in fruit flies, and wing shape can be normal or vestigial. Now you just did a fruit fly lab where one of the crosses body color and wing shape. And you should have noticed in that cross that those are linked. Those are genes for two different traits that are linked on the same chromosome. So let's say that we did a cross with a heterozygote here. Let me get my pen going. A heterozygous fly is crossed with a homozygous recessive fly, right? Very similar to that F1 cross that you did in your lab. And this was the data. 778 flies were born with gray body normal wings. Now that would be gray body, a capital G, and a capital N. So both dominant traits are showing here. Black body vestigial rings would be 2 little g's because black is recessive. Vestigial is also recessive, so 2 little n's. So pure recessive, 785. Black normal would be, okay, 2 little genes, recessive black, but dominant normal wings. Gray body vestigial wings would be big G for gray body and little n's for vestigial wings. Okay, so definitely this does not follow the 9:3:3:1 ratio. And it wouldn't anyway. You would actually be expecting if you did the Punnett square for this, let's do the Punnett square. This individual here would only be able to make alleles with GN, right? So I'm going to put that parent over here. That's the only gamete that individual can make. And this individual, if we foil him, right, or her, you get the classic, you get big G, big N, you get big G, little n, you get little g, big N, and little g, little n. And I didn't draw 16 boxes. You don't need to, because this guy, there's only one type of gamete. So you should actually get a 1:1:1:1 ratio here. And you did in your lab as well, by the way. So here we get big G, big N, little n. So both dominant traits, you get 25% or 1. With this one, we got big G, and 2 little n's. So dominant recessive trait is another 1. Again, following Mendel law, this is what you would expect, right? 2 little g's, a big N and a little n. So that recessive, that dominant, another 1. And then all recessives, we expect it again. And really, it's 25% each. You can also say it that way. Or you can say 1:1:1:1. That is not what we have here. Look at these crazy numbers. If we were expecting all of these to be 1:1:1:1, we didn't get that. We've got 780, 150, 150. That's really off. This definitely tells us that we have linkage here. So now, in order to calculate the distance, right, between the genes on the chromosome, this is the formula. You have to count the number of offspring that show recombinance, that are showing recombinance, or evidence that they crossed over, right? Divided by total number of offspring. Total offspring that were counted in that experiment. Total, okay? So number of offspring showing recombinance, what exactly does that mean? Well, that means they're the number of offspring where if you had, if you had the chromosomes lining up during meiosis, and I've drawn this before, and let's say that your big G's are up here, and your little G's are here, and your B's ends. Okay, put ends down here. Ends and little ends, little ends. How many times did this guy cross over and have these guys switch places? So switching places would make this allele and this allele. That's the only way to get those alleles and therefore get this type of offspring here and this type of offspring here. The only way to get these is by crossover events. Crossover events are what are creating those offspring. So black body normal wing is a recombinant offspring. So is gray body vestigial wings. So these two right here, the two numbers that are a little smaller than the other two, usually that's how it works out. Those are the ones that are recombinant. Those are the recombinant offspring. Recombinant offspring. Okay. So what do? We add them up. We put them into the formula. 158 plus 162. Of course, I put my key away. Okay. $158 plus 162 comes to 320. So 320 is on the top of my fraction, and on the bottom, I believe it is 1,883. Total offspring. If I total all of these guys, correct me if I'm wrong, guys, but I'm pretty sure that's what that is. Okay? So there it is. And that comes to 16.169. So then the next thing that I have to do is I have to multiply that by 100. And that gives me 16.9, which is my crossover frequency. That is my crossover frequency, or the frequency of offspring showing that they resulted from a crossover, right? Okay, so you also just very simply translate that into a distance. That's exactly what we're going to have you do, is we're going to say, all right, now you're going to draw the chromosome. If I can get this over here. No. Come on. There, okay. So now the chromosome, if you were going to draw it, which is exactly what I'm going to ask you to draw on the Fly Lab, if I were going to make a distance between, here's my G's and here's my N's, this distance on the chromosome is 17 map units long. You can round it to a whole number, that's fine, okay? All right, so that's how you calculate your map units if you're given the offspring data. The other thing that I'm going to add to this video, which is also really useful, is there's kind of like a fun sort of a puzzle that they'll sometimes give you. Sometimes they won't give you offspring data. Instead, they'll do something like this. Number 4 is a gene mapping example. So they might give you given crossover frequencies between pairs of genes. So not just 2 different genes, but maybe 4 or 5 different genes, and they'll maybe give them letters like A, B, C, D, E, or something like that. So for example, they'll give you A and E crossover 8% of the time. W and G crossover 15% of the time. W and E crossover 3% of the time. So you always want to go and start with the ones that are furthest apart. That's how I do this. So 15 is your biggest number. Convert percentage into map units. They're the same thing. So I'm just going to draw a line and say that this is my chromosome. Here is my W. here is my G, and this is 15 map units because it crosses over 15% of the time. They're the furthest apart. Okay, then I'm going to go to this one because it references a gene that's already on my map. W and E, a new gene, cross over 3% of the time. So I'm going to go, okay, right about here, I'm going to put E, and I'm going to say this distance is 3 units apart. And now I just need to add the letter A. So where would A be on my map? Well, it's 8 units away from E. So you have a choice. You could put A over here and say that this is 8, and that would be a right answer. That would be fine. You also could put A on the other side of E. You could put it over here since it doesn't tell you how far A is from G, right? So you could lengthen this and go A and E, and then this would be a 5. So this is an example of a mapping where you could get away with 2 right answers. Okay. But it's kind of like doing a puzzle, and it's kind of fun, and you might get something like that next Tuesday on the paired essay. And that is all I had to explain to you, and I hope that was helpful.